Hello, everyone, and welcome to PyTorch Meetup number 10. It has been an exciting journey so far, and it's incredible to see so many of you here joining us today. And we'll have 3 speakers representing both more academic side and more industry startup stuff. And we also, for the first time, have a speaker joining us from a different continent, from California. So since we were not able to have in-person events for quite a while now, so every time I keep thinking, what's something we haven't done yet that I could try to pull off? So last time it was a collaboration with a different organization, and this time I decided we could make the most of not being in person and have a great speaker from a different side of the world. So yeah, I'll hand over to Paul to say a couple of words about how he feels about the past 10 events we had, and then we'll get on with our first speaker.
Well, I'll be very brief, but I'll just say thank you very much to everybody for being so loyal to us, and I'm glad that the group has been growing at a nice pace. And really, I would just like to particularly thank Karina, because she's the one who kind of held it together throughout the whole Corona crisis and everything, and kept it going, and hopefully will return even stronger after the crisis, and have some more offline events and share some more beers together. So that's all I want to say. Thank you very much.
Yeah, sharing some beers together will be nice as soon as possible. I keep moving the booking in Microsoft Reactor for next month. So one day it will actually be allowed and it will happen.
That's great.
All right, Doug. I think that's the majority of people who wanted to join already here. And it's 6:40, so I guess it's a good time to start.
Okay, let me try and share my screen. Okay, can you guys see?
Yes.
Okay, awesome. So I'm going to be presenting some research on 3D reconstruction. This is work that was done at Magic Leap. And I'm only going to be talking about the research that we are presenting at ECCV later this week on Thursday. I'm not going to be talking about anything Magic Leap company or product related. So 3D reconstruction is becoming important in a lot of areas. Some of the most noteworthy are autonomous driving, where the car needs to understand the 3D world around it so it can navigate safely, as well as in augmented reality. And on the right, you can see the Magic Leap goggles. And on the left, I'm going to play a snippet from this video showing a cool new feature from the iPad Pro that comes enabled with a LiDAR scanner. So I like this. So I like this because they're showing their LiDAR scanning app, and actually some of their visualizations look very similar to visualizations that we'll see from my method later. But we can see that Apple has invested a lot of money on adding depth sensing LiDARs to their hardware so that they can do 3D reconstruction, things like this for augmented reality apps. And so here you can see they're reconstructing a 3D model of the world in real time on the iPad. So in this talk, I'm going to give a little bit of background on active depth sensors, and then go on to passive visual depth prior methods, and then some depth fusion. And then we'll get into the work that I did, known as Atlas. And then I'll talk about some related and future work, and then conclude. So active depth sensors are used for getting high quality depth. And here you have not just a camera sensing the environment, but you actually have some sort of illuminator illuminating the environment. And this helps solve for the depth. So in LiDAR scanners, you shoot out a laser beam and you measure the amount of time before it bounces back, and that gives you the distance to the object. In structured light, you have some sort of projector that projects a distinguished pattern onto the world, and then the camera observes that and can triangulate more easily using that. And these methods work very good when they work, although they fail under different, when the illumination is too bright. So things like Connect doesn't work in outdoors very well, although LiDAR scanners now on cars are bright enough that they work. Another drawback is it requires expensive custom hardware, whereas cameras are very cheap. And so work on depth estimation using just a single camera. One approach is monocular depth, and this is kind of the most straightforward, simplest way to think about it in the era of deep learning, is that you have a single input image, you pass it into an encoder-decoder network, and the network then regresses a depth map. And here you can see some results from one of the state-of-the-art methods. And they have the input images and the colored depth maps. And so while these look pretty nice, what I found when I started playing with them is that the depth maps look good, but when you actually look at the 3D point clouds that are generated by them, they're not actually correct metric depth. And so these networks are almost more learning kind of like to segment depth planes than to really get high quality depth. And so if you try and fuse these into a full 3D model, they don't work very well. Sorry, can you guys still hear me? It's silent on my end.
Yes.
Okay, cool. So the next way of doing depth estimation is to use binocular or multi-view. And here you have multiple cameras observing the same scene. And these multiple cameras can then triangulate points because you know the difference between the two cameras. And so you have rays that intersect in the real world. And this resolves a lot of the ambiguities that are present when you only have a single view of the scene. And so here there's a bunch of recent work, all kind of have their own twists on the problem or the solution. But the basic idea is very similar. And that is you take your image, you take a pair of images, you extract features from both of them, and then you warp the features into a 3D cost volume where each warping corresponds to a different hypothesized depth plane. And so you get a stack of different images as if it would like of one image projected onto the other image as if the whole scene was at that depth. And then you have a 3D network that takes this volume of images and processes it and regresses a probability, which can then be flattened into a disparity map that gives you the depth. There's also these methods kind of mirror previous classical approaches, where instead of deep networks learning these functions of how well pixels agree in the different depth planes, they had classical measures of agreement between pixels. So then once you have your depth maps, the standard thing to do is to fuse a bunch of different depth maps into a 3D mesh. And so a very popular approach is TSDF fusion that is often applied on depths that come from some sort of depth sensor, like the Connect depth sensor, but can also be equally well applied to depth maps from monocular or multi-view stereo methods. And here the idea is that you discretize your space into a voxel volume, and each voxel stores the distance to the closest surface. It gives you a little bit more information than just storing whether the voxel is occupied or not, but you can kind of think of it as a occupancy probability. And so then the surface is extracted by finding either where the voxels are occupied, or in the case of a TSDF, where the zero crossing is. So the distance to the surface is 0. And so by putting all of the depth maps into this voxel volume, you can average them together and reduce noise and fuse them into one coherent 3D model. And so when I saw some of the results from these deep learned stereo methods that we saw on the previous slide, these results actually start to look pretty good. And I thought that by combining this with some TSDF fusion, that we might be able to generate high quality 3D models just from a single camera moving around an environment. But what I found was there was too much, the quality of the multi-view methods still aren't good enough that when you fuse them, you end up with not very good looking 3D models. And so that's where, so then another approach is once you've fused the 3D model, there's some work that takes a noisy input TSDF reconstruction and passes it through a 3D encoder decoder network. And this network learns to clean up the geometry and improve it, so to reduce noise and fill in the holes. So I was like, well, I can try retraining this to handle the noise characteristics of the multi-view depth map input, and maybe that would work. But along the way, I realized that there's kind of, this is a roundabout way of doing it. Now that we have an encoder decoder network that is regressing the 3D structure, it seems kind of unnecessary to generate these intermediate depth map representations. And really what we want to do is take the input images, feed them into a network, and then feed all of the images into a network, and then combine all the information, and then directly regress the 3D TSDF end to end without having to go through the intermediate depth maps. And so that's where the Atlas work comes in. So here our network architecture is, we have input is a bunch of just standard images, like RGB images from a camera. Each image is passed through a 2D CNN to extract features. These features are then back projected into a voxel volume. And then all of the features from the images are kind of accumulated together. We use a simple running average, although I think that in the future, other types of accumulations, maybe like an RNN, could improve the results. But then once the features are accumulated in 3D, we have a 3D CNN that then regresses the final TSDF. Other things such as semantic labels. I think other interesting things to regress could be like intrinsic image decomposition, texture, but in the paper we only tried with semantic labels. So now I'm going to play a short video giving a bit more information about the method. And oops, can you guys hear the video when it plays?
No.
Oh, you don't hear the audio?
It's not moving at the moment.
Oh,
it is
is
now,
it?
but no audio.
Okay, so I'll just try and talk over it. 1 second. Yeah, so here we see a video and the 2D features that are being extracted. And then these 2D features are back projected along rays into a 3D volume. And here you can see the volume being filled up with the features. Then once these features are accumulated, you can run the 3D convolutional network to regress the TSDF function. And then finally, you can extract the mesh using marching cubes. You can also get semantic labels. So here we're comparing our results to 3 state-of-the-art methods: GPMVS, DPSNet, and MVDepNet. These are some of the binocular methods that are multi-view stereo methods that I was mentioning earlier. And you can see that the results are very noisy. Although some of the information is there, the temporal consistency creates a lot of noise that isn't removed by the TSDF fusion. So here I'm showing the results run incrementally and overlaid onto the scene. And this was the visualization I said looked very similar to the one that Apple is doing on their iPad. But here we don't use a LiDAR scanner, we're only using a standard color camera. And the colors of the objects represent the semantic segmentation. So green is floor, yellow is chair, walls are blue. So I'm not sure how well that video played when it was shared as a screencast, but I suggest you go and watch the video at full resolution and quality with the original voiceover, which was better than my voiceover just now. So moving on some of the details of the results. The inference time, we compared inference to some previous methods. Here, the previous methods are only regressing to the depth maps, and I'm not including the additional time required for TSDF fusion. And we can see that our method then, we run the 2D CNN on each frame to extract the image features. And these features are then averaged into the volume as like a running average. And so we only need to run the 3D CNN every time we want to output a mesh, often at the end of a sequence if it's an offline reconstruction, or as often as you want to see a new mesh appearing. So we can accumulate information in real time or close to real time, but we can only output meshes currently at slightly better than once per second. Although I think that with some optimizations and this can be reduced quite a bit. And so we see that ours, when you average over, like when you amateurize the cost of the 3D CNN running over the scene, our network ends up becoming competitive, if not a lot faster than some of the previous methods. Another cool thing that our method allows is the completion of geometry. Here I'm highlighting two different reasons why geometry might be incomplete. If you're using a depth sensor, then you have the problems that are apparent in highlighted in A in the purple. And that is that depth sensors work well on kind of diffuse materials, but on very dark or very specular and shiny materials, depth sensors start to fail. And so you end up with holes in the mesh, even when in the ground truth that we consider from the depth sensor. The monocular and binocular methods don't have this issue. They predict depth for every pixel, but the results are often not great. And then in addition, there's this other type of missing geometry shown in green in B. And this is due to having never observed that portion of the scene during the reconstruction. So it's kind of an area that was occluded, or you didn't aim your camera there. And none of the previous, basically, if you're doing straight TSDF fusion, there's no way for it to inpaint this information that wasn't observed. The one paper that we saw that kind of trains a 3D encoder decoder to reduce noise and fill in holes actually does a really good job of filling in these holes. But our method just directly does this implicitly. our method also is pretty robust to the number of frames used. So here I'm showing results if you only use 25 frames from the sequence, 50 frames from the sequence, or all 800 frames from the sequence. And you can see that even with only a relatively few number of frames, the results are still almost indistinguishable from the final result. And an interesting related work that is also being presented at ECCV this year is NERF, neural radiance fields. And here they have two very interesting ideas. One is that instead of representing the volume as a discretized voxel grid, they represent it as an implicit function where you have a multi-layer perceptron that takes in the coordinates of a position in space and outputs the information for that point in space. And so instead of having to discretize voxels, they can evaluate what the function will be at any point in 3D space, allowing for arbitrary resolution. And then the other very interesting part of this work is that they train their model by then doing differentiable rendering of the volume. And so this allows them to train their method just on images without ground truth depth for supervision. And they're able to produce really nice results. So here you can see that they're doing novel view synthesis of this kitchen scene. And so all this video of moving around is all the network rendering new views of the scene from some few input images. And you can see that it's even capturing how the specular surfaces reflect light differently depending on the viewing angle, which is often not captured in a lot of other 3D reconstruction methods. And so I think it'll be very cool. Oh, the problem with this method is that it currently requires close to or over a day to get the results. So if you have a set of images, it takes like a day of gradient descent in order to get these results, whereas my method runs pretty close to real time. And so I think interesting future work will be to combine ideas from both papers and use implicit functions for regressing the TSDF so that we can create higher resolution, as well as training with this differentiable raycasting rendering that will allow for self-supervised training and training on a lot more data and potentially creating really high quality visual results like these. So that's the end of the talk. Stay in touch. My website is zach.murez.com. The paper and code can be found on zach.murez.com/atlas. And I hope to be able to create a smartphone relatively live demo where the smartphone can connect up to a desktop GPU server in the cloud. We'll see if I can actually get that to work. Also, come visit my poster at ECCV on Thursday. And thank you. Do you have any questions?
Thank you so much, Zach. That was an amazing talk. I learned a lot. And yeah, everyone, if you want, you can put the questions in the chat or just unmute yourself and jump in with the questions.
In terms of the hardware, what processing platform are you using?
Yeah, so the networks are all trained, and I've only so far done inference on desktop GPUs, so like NVIDIA, big beefy ones that they top of the line ones. I think we used like the Titan RTXs or something.
So this is not applicable for robots, or I mean, slams, stroke point cloud in real time.
Yeah, so I think I see two ways forward on that. One is this was kind of an academic paper, and so I didn't spend any time trying to apply the techniques of optimization to the network to make it much smaller. So like quantizing the weights or shrinking the model. So I think those can all be applied and probably get pretty significant performance increase.
Okay.
And then the other direction is, I think that Wi-Fi or wireless kind of communication is often available, in which case you can use edge or cloud compute. So that was the idea for my iPhone like demo is I don't want to, I think it'll be more work to actually get it to run on iPhone hardware, but most of the time iPhones have internet connection and can transmit frames to the cloud and then receive the mesh back. If you wanted to like actually make a product out of this, you'd have to worry about privacy and all sorts of things like that. But as just like a fun demo that people can download the code and run themselves, I think it's possible.
Thank you. Cool.
So there's a question in the chat saying, what made you choose PyTorch Lightning?
Sorry, what?
What made you choose PyTorch Lightning instead of other?
Oh, why did I choose PyTorch Lightning? Yeah, I kind of ran across PyTorch Lightning and saw that it had a whole bunch of nice engineering already done for me, particularly the distributed data parallel training that allows you to train on multiple GPUs, as well as 16-bit floating point precision stuff. Both of those are kind of a lot of engineering effort to set up and get running correctly in just plain PyTorch. But in PyTorch Lightning, it's all handled for you. And that was the main reason. And I think that before I switched over to PyTorch Lightning for this project, I was always training my models in 32-bit floating point. And I found that with 16-bit for deep networks, it's kind of, the results are exactly the same, if not better. And so there's no reason to waste the extra compute and memory. I was actually able to, like the trade-off between the less precision didn't really matter compared to being able to have almost like double the batch size. And so the training was much more stable, actually using the lower precision. They also have a whole bunch of nice, all the rest of the boilerplate training code, so you don't have to write your own training loops and evaluation stuff. So it makes starting up a project much quicker.
I see. So while we're waiting for more questions, my question would be,
okay,
sorry.
You can go. No, please go, Karina. I'll ask after.
So my question was, what part of the project took the longest time to get working?
Well, I guess the hardest part was coming up with the idea, the realization that this can be done. But that wasn't necessarily like time that I was working. It's just kind of, that's the nature of research.
Yeah.
After that, I would say probably debugging some of the 3D geometry stuff. That when there's subtle bugs in the way that you back project your information into 3D or do those kind of ray steps, the network is actually pretty good at learning to correct those mistakes, but the performance isn't nearly as good as if you actually get those things correct. And so you get results that are like kind of working and you're not sure exactly, is it the network just can't learn more? Is this like just as good as the method is going to be? Or is there some sort of subtle bugs that don't cause runtime errors, but are just not correctly implementing the math of the 3D geometry? And finding those is the hardest time consuming part.
Yeah, it was the same in my research when you have no idea if it could be better or is it just as good as it's gonna get, and
yeah,
think of structurally completely different approach. All right, so Roland, did you have a question?
Yes, is more towards Magic Leap, if that's okay. So, considering the recent transition to enterprise and more towards applying the headsets in the industry, wanted to ask you if the research there has changed, or do you have like different priorities, or what's the main focus now in the research side?
Yeah, I'm just going to talk about the research being presented here and not discuss Magic Leap's priorities going forward. So sorry, I can't answer that.
I see. It's all right.
Do we have any other questions? Looks like no. All right, well, thank you so much for this amazing talk. And
okay, I'm going to stop sharing my screen.
Thank you so much for joining us from far away. It was our first time inviting somebody from a different continent, and it's really nice to be able to do that while we're unable to do other fun stuff in 3D world. So yeah, thank you so much, and yeah, please do join us for the rest of the event, or if you have to run for the conference, then we really, really appreciate your contribution. Thank you.
Yep, thanks for having me. I'll stick around for as long as I can.
Amazing. All right, it's now a talk from Artur about pathogens versus machines.
Thanks a lot, Karina. Can everybody see my screen and hear me?
Yes.
I know it's a classic question these days. Thanks a lot for having me here. Hi, everyone. My name is Artur. I'm a co-founder of a not-for-profit organization called Artificial Intelligence for Life Sciences. And I'm going to start actually my talk with like a portion of shameless ads. Hope that's fine with everyone. Basically, I just wanted to showcase what we do at AILS. AILS is essentially a platform bringing together people who are willing to contribute to revolutionizing life sciences through artificial intelligence and deep learning. And we are kind of free-floating platform and a discussion forum. We run as a forum, essentially, bringing people together in answering research questions, doing journal clubs, sharing papers, sharing code, and all sorts of things. And you know, if you like a little bit of the direction of stuff I'm going to be talking about today, come join us, share some ideas. We would love to have everyone who is engineers, who is researchers, who is just, you know, a citizen scientist, if you will, who is trying to understand what's going on right now in life sciences and deep learning. All right, so with this, I probably would start the short introduction to the problem setting that I'm going to talk about today. And specifically, I'm going to talk about host pathogen interactions and how deep learning and artificial intelligence can be used in this setting. And a lot of the work that I'm going to be talking about today is done either as has been done some time ago or as a part of AILS, or as a part of UCL, or in collaboration with Francis Crick Institute. So essentially, host pathogen interactions and what we do with this, it's a very timely topic because obviously every one of us is now locked down a little bit. A lot of us are working from home. We just got out of a major lockdown in the UK, and you know, hopefully not going to get into another lockdown. But basically, let's talk a little bit how research in pathogens is done in essence. I think this is a very minimalistic representation of how this can work. And quite often, if researchers find new pathogens, they will try to isolate them and culture them in the lab so that they can do experiments with them. And while they do that, while they work with the new sort of pathogens, they try to develop some molecular tools that will allow them to work them, to visualize infection, and to visualize how these pathogens proliferate in for example, cells or human cells. And all this body of work usually leads to discovery process of mechanisms that, or more sort of focused on utilitarian application, could be antivirals and vaccines. And after that, there is also some post data acquisition analysis that is done on this data. What we were doing is we're trying to bring more of this analysis directly into this discovery process, and sometimes even to an early tool development phase to kind of try to enrich this part of the pipeline. So first I'm going to talk about some throwback work that has been done some time ago together with Francis Crick Institute. And this work was on Toxoplasma gondii, a rather successful parasite that looks like shaped a little bit like this. I think it's an artistic representation. That was done together with a group of Eva Frico, who was at that time at Francis Crick Institute. And probably some details of what you need to do to know about the Toxoplasma parasites and infections by Toxoplasma is that Toxoplasma can infect essentially any nucleated cell, and it will establish own vacuoles, so some sort of compartments within the cells where it will proliferate, and eventually the cells will burst. It will release infectious parasites around that would go and infect other human cells. And this is an unfortunate event where an infection by this parasite would go wrong. However, this can be prevented by the cell itself, by an immune system, an immune response to this infection that would be occurring due to the so-called host pathogen interactions. So the parasite itself will try to mark these parasites with proteins, and then the other proteins would be responsible for detecting this markers and killing the parasites essentially, and the cells would go on and live happily ever after. So before I go into specific details of the project, maybe I'll explain a little bit how this kind of work is done in the field. So quite early on, imaging came and digital imaging came into this kind of work, using quite often as well automated microscopy techniques. And what you're looking at here right now is a merge and individual channels of fluorescence micrographs, so microscopy photographs, if you will, of individual cells cultured on a dish surface, where cells are marked with fluorescent dyes. Here is a dye for a nucleus, the blue one, and the green one is a dye for the pathogen. Well, it's actually not exactly a dye, it's a fluorescence protein that this pathogen is expressing. There's also a dye for the cell outline, and you can see here that the whole analysis is well separated already. You have separate channels that you need to worry about segmenting in some certain way. So fluorescence microscopy is quite an interesting technique that it uses scientific imaging at the start, at the core by design. And using this imaging techniques, obviously there is a lot of sort of heuristics involved in analyzing those images. Way before the sort of advent of complex analytical methodology, scientists were capable of counting the cells automatically using rather basic thresholding techniques. And unfortunately, in some cases, so here we could look at the nuclei, detect the nuclei. Here we could look at the pathogens, detect the pathogens. Here we can get some idea of the cell outline to kind of match together the nuclei and the cell outline, and trying to match whether the pathogens are inside of the cells or outside of the cells. However, as you can see here, quite often this standard thresholding techniques would be not precise enough to detect individual parasites at a higher resolution when you want to push it a little bit more. And this is exactly where applications like machine learning could come in and help out to quantify the parasite load and try to understand where the parasite is dividing or not, and what's the situation in terms of infection in this cell. So here we used rather simple decision trees, and that turns out is sufficient to understand what's going on in this specific pathogen. However, in situations where we want to actually understand whether there is an interaction between the proteins of the cell and the pathogen itself, things get a little bit more blurry, as you can see here. It may be, if you're a classical microscopy image analysis person, you'd be like trying to get an idea of whether there are the magenta and the green is somehow overlapping based on the magenta intensity. However, quite often, due to the quality of these images, this will not be really possible. And what you actually are looking for situations like this, where there's a coat of protein around the pathogen. This is a clear situation that this pathogen would actually be cleared for being removed from the cell. And this is exactly the situation that deep convolutional neural networks are good at detecting. And again, this is a little bit of a throwback work. So you can see that here we used an architecture akin to AlexNet. Obviously, also we're a little bit limited in the amounts of data that we could apply for this work. So AlexNet worked really well for us to detect this recruitment phenotype. And what additionally it gave us an idea to understand, to sort of use this implicit unsupervised component of deep neural network to understand what is it that the network is looking at when it's looking at the pathogen recruiting the host proteins here. And for whatever reason, this is something that would have to be followed up by the biologists, hopefully. For whatever reason, the neural network was looking at the tail of this pathogen here. So the take-home messages for this work are that actually the Herman, the framework that we developed here, was an open source user-friendly software for host pathogen interaction analysis on a single cell level. It was designed for biologists and did not require any coding, based in the sort of a background, if you know software called Nime. This is what it was based on, with a lot of bells and whistles on top that we developed. And basically, through this work, we were able to convince the community that they need convolutional neural networks to do their image analysis, and that it's quite useful to have those techniques at hand. Now, let me go to the next work. I'm going to be racing a little bit here. I'm just going to give a broad and introductory talk of all kind of things we've been doing. And this work was done in a collaboration with University of Zurich, a group at University of Zurich, and the first author of this work should be on the call, I believe. So he would help me out, I hope. In this work, we looked at two virus infections, herpes simplex virus 1 and human adenovirus. Quite different viruses, quite different biology behind that. I'm not going to go too much into detail. I'm happy to do that afterwards. And essentially, we went a little bit earlier, if you remember the pipeline of the discovery that I mentioned before. We went a little bit earlier into this pipeline, trying to bridge sort of or attack the point before the amazing and great molecular tools like green fluorescent protein, genetically modified viruses encoding green fluorescent protein marking the infection cell that you can see here. Before these tools are developed, can we actually look at this green signal, this wonderful green signal, then take a very basic dye for cell nuclei off the shelf that's probably available in every lab in the world, and use the nice and beautiful GFP signal. Those two. So again, those are two different channels of the same image, essentially, of the same site. or the same field of view. Can we take this as the ground truth and take this as a signal and essentially train a neural network that would be able to detect infection without GFP at all? And turns out that using a ResNet-like architecture, you can do that. And you can do that very successfully for both adenoviruses and for herpes viruses. And you could do that for viruses that encode GFP, or the viruses that are so-called wild type. That means they're like completely isolated, the way they are isolated from the wild. And the ground truth here was obtained from so-called immunofluorescence, or also known as basically an antibody staining. And we have shown here that using these techniques, we were able to combine these 2 points and be essentially able to create a classifier substituting the specific signal. Okay, so beyond that, there was a very interesting phenomenon that we observed, that a lot of adenovirus-infected cells were not lysine, go into the end of the life cycle of the virus to release the progeny of a newly synthesized virus. And it turns out we were able to predict, also using similar techniques, that there were changes in morphology of the cellular nuclei that gave us information about whether the cell will lyse or not. And this brings me again to the take-home messages. ResNets can be used for biomedical images. This is great. Not the most exciting news anymore, I think, as the whole field is getting more and more revolutionized. But what is very interesting is that biological markers can be efficiently used as ground truth, and we can train machines to detect complexity of the morphology that would otherwise be ignored by a human eye. And this brings us to a creation of quasi-label-free microscopy, which is pretty cool in my opinion. And obviously, additionally, we can obtain some mechanistic information of the way infection develops and it goes on. Now, for the dessert, let me talk a little bit about about smallpox viruses, a virus that is eradicated, but back in the day, it was responsible for killing some 300 to 500 million people, and luckily it was eradicated. However, today, due to the fact that the vaccination campaign is no longer, human population is susceptible, and certain flavors of the virus, essentially like monkeypox and sheeppox and goatpox, still represent a biomedical and an economic problem. So here we used deep learning on a completely different scale. We looked at the subcellular resolution using a fluorescence microscopy technique called super resolution microscopy, and we have devised a sort of a method to give us a little bit of a shortcut for obtaining Z profiles in 3D of virus particles. Again, conveniently, they are separated in different channels, and you could finish the story here essentially and do nothing more and say, okay, we just use the fluorescence channels that is already separate to just understand which different flavors of particles do we have there. However, something that you cannot do with this kind of techniques is try to understand whether viruses are changing depending on whether they go into the cell or they're outside of the cells. And here we used several different architectures, and most sort of interestingly, we also used capsule networks. which we were initially unable to train on this data set. However, rather early we realized that the data set that we've got in our hands is remarkably reminiscent of the, yes, I know, MNIST, the length we go to keep using MNIST dataset. So it turns out that if we slightly modify our dataset to look like MNIST, we could do the transfer learning successfully and train successfully a capsule network on this dataset. Which was not possible to do without previously training the data set, the MNIST, the capsule network on the MNIST data set. Luckily, the MNIST allows us to train CapsNet, and it also allows and train CapsNet allows us to use the generator part of the CapsNet to see what are the differences between the cell-associated and cell-free particles, and we can. This is completely new to virology. This is something that was not really observed. Again, just because we're talking about really a lot of virus particles, and no one would have the patience to sit down and try to summarize you know, the minute differences between particles inside and outside of the cell. And this is something that neural networks can provide us almost for free. And finally, we were able to perform similar application of the mechanism that we called mimicry embedding, basically using MNIST dataset to pre-train different architectures to be able to detect dead or alive toxoplasma in their cells. And we were even able to do that in vivo in an organism. And that's probably the final take-home messages. Here we were able to focus on subcellular structures. And just because microscopy is usually revolving around, you know, sizes like cells and or full organisms, immediately you'll get a larger size of individual examples of your pathogens, and that boosts your biological data set a little bit. We use mimicry embedding as an efficient way to facilitate transfer learning from seemingly unrelated data sets. And also bringing it into this very standard MNIST shape allowed us to just throw any architecture that has an implementation for MNIST, or any kind of method that has an implementation for MNIST, which is quite convenient. And finally, deep learning can be used to detect subtle patterns in pathogens and refine pathogens analysis. That brings me to my acknowledgements. I'd like to thank Mercer Group, formerly at UCL, currently at University of Birmingham, Frickle Group, and Grabber Group at the University of Zurich. And Vardan, I think, again, is here on the call, so maybe he can say hi later on. Yeah, so that brings me to the end of my talk, and I'm happy to take questions.
Thank you so much, Artur. Do we have any questions? As usual, just pop them in the chat or speak up.
Yeah, I do have a question because you use the handwritten recognition, MNIST, and all those static data as sampling and data set to look at a dynamic environment. So I was wondering, pathogens are dynamic, non-stable, non-static. So if I'm going to collect information about cloud, about, moving waters, I need to be able to sample for a period of time. So why MNIST?
Fair enough. It's a very good question. I actually, I guess, by having a background in infections biology, this never really occurred to me in the same sort of way. I guess you wouldn't compare really pathogens being dynamic as much as clouds are, just simply because pretty much every pathogen in the world follows a very strict genetic program that is encoded in its genome, and at any given time, essentially, you quite often use pathogens actually to time and synchronize certain cellular events that you want to look at. They are so this life cycle of a pathogen is so sort of straightforwardly predictable and deterministic that it's a remarkable tool for cell biology, and it's quite widely used, this concept. In fact, yeah, so all the pathogens are strictly encoded by their genome, and the program unfolds from the moment they cross the cellular membrane and they infect the cell. So in this sense, if we do snapshots in time measured in a very strict moments of time after the infection is happening, we're looking at quasi very similar entities, if that makes sense. And I think it's very difficult to do this with clouds, but you definitely can do this with pathogens.
Oh, okay, so it means you're looking for common denominators.
Yes, and in this sense, time after infection is usually the most relevant common denominator across the pathogens.
Okay, thank you.
Any other questions?
Yeah, I had a question. So I was curious what your impressions are for capsule nets. Like, did you enjoy working with them? Do you think they really are gonna take off and work? And kind of specific to that, I was curious that you said that it wasn't able to converge when you kind of trained it from scratch, but that transfer learning helped. I was wondering, does that mean like your data set that you were targeting was just kind of too small, or were there other stability issues that just made it really hard to train?
That's a very good question. Yeah, so I think capsule networks are tricky beasts, to be honest. And they behave quite odd quite often. So in the sense of training, we basically had a little bit of a stability issues of trying to train them on non-standard data sets. So on things like MNIST, they would eagerly converge and things will just happen automatically, if you will. However, whenever you would bring, let's say, a real-world data set, things, highly imbalanced, even, if you try to use generators and so on to make sure everything is more or less stabilized in this sense, they would simply not converge, and that was a very painful experience, and that's why we decided to go for transfer learning straight away, and that worked surprisingly well. Yeah,
it's surprising that like there's enough pre-training information in MNIST for it to actually do anything. Like ImageNet is like a much larger, more diverse thing that's used for kind of transfer learning, but it's cool that MNIST has that information in it as well.
Yeah, and I guess it's also the sort of a nature of the data set being very close in terms of just pixels and the grayscale values. And MNIST as such is, nicely polished, and also it has enough. So I have a feeling that it just has enough challenging things to look at for high expressive capacity architecture like CapsNets.
Okay.
Yeah. Well, hopefully it goes the direction of GANs, where they started off being beasts, like you say, but eventually they're being tamed.
Yeah. So I think there's, I'm kind of talking just like completely speculations right now, but I heard about quite interesting things being done in the domain of universal representations, and maybe something like this could help sort of tame the CapsNets after all.
So do you have problem explaining, explainability of this and interpretability? Is there a problem there to see whether this is the pattern you are looking for? This is
regarding this, I think CapsNets are actually very good in terms of they have the generator sort of built in.
Yeah.
So you can immediately look at what is going on in terms of representations that we're looking at.
Okay. Cool. Interesting story. Very interesting. Cool stuff.
All right. Do you have any other questions? Your last chance. Jump in if you have one.
Yeah, very interesting. Very interesting area. So looking at bioinformatics and so on.
Yeah, so please do join us at the ELSTOT Institute, and we're happy to continue. I'll be happy to continue conversation there.
Yeah. Good. So, yeah. Hello.
So, Artur, I have a question that is not exactly a scientific question, but more generally speaking, when you're trying to develop a machine learning model that can be used by somebody who doesn't know any coding, like biologists in the lab, I guess it's quite a big challenge of how to make it user-friendly in a way. Because when I say user-friendly, I mean command line friendly for everyone who is comfortable with coding. But how do you make it user-friendly for somebody who has completely different ways they interact with computers, and how to make them trust it and want to use it and be able to make the most of the results? I guess it's a very open-ended question, but
yeah, I see what you mean. So I guess in this sense, I had really good experience with sort of graphical workflow platforms like Nime or I think, what was it again, the Taiku or something like that. So somewhere where a biologist could see sort of a flow chart of what's going on there. And, I would just package all the heavy code and deep neural network inside of one of those nodes, and it just would be a black box to them, and they would use it. However, what I think is usually a really good idea, if there is something that you want to, that you might not be always able to support in a really long run, let's say over years, let's say things will just deteriorate, the data something will happen to the data baseline and so on. I think what is usually a really good idea is to, instead of just doing end-to-end deep learning based solution, that have sort of a hybrid solution with a little bit deep learning based for better features and better, let's say, accuracy, and a little bit of a rule-based part of the framework. And this is exactly what we did with Herman, for example. We did a combination of rule-based and machine learning-based solution. So basically, if the machine learning-based part fails, the rule-based part will pick up and provide at least some readout, and also provide a sort of alternative to be able to benchmark the results that you will get from a deep learning system. So, and I think this is, it might feel intuitively, if you do a lot of deep learning, it might feel intuitively as a little bit of a step back. However, I think it's a very robust way to build systems.
That's a really awesome answer. Thank you, Artur. All right, if there are no more questions, then we'll move on to our third talk by Peter. And this talk is very close to my heart because it's about waste management and how we can improve recycling. So for those of you who know me, you'll know how much I care about this talk for my personal interest. Thank you so much, Peter. The screen is all yours. We can't hear you yet.
Cool. There we go. So now I'll share again.
Yes, please.
Okay. Is that working, Tom, when you can see it?
Yeah, go for it.
Okay, so I'm coming from the more production side with this talk. So I come and I'm the CTO of a company called Recycleye, and my slides work. What we do is waste data and analytics. So when you take your can or a PET bottle and you throw it in the bin, that's not the end of its journey. It gets picked up by a truck, it gets transported to a waste facility, and then it goes along a number of conveyors and it gets sorted generally by some large machines and also by people. And what Recycleye does is we take computer vision and we apply that to this waste data. So you can see, which I don't know whether you can see the thing, but we have edge devices which are running on top of conveyor belts. And then we are generating reports and data for clients that allow them to see metrics on things like purity, contamination rates, and see the trends over time of their waste batches. So they can start tuning machines in the plant, and they can have a much better idea of the composition of their waste. So back to the start, and I think this is a nice progression of how Recycleye started and also how our data journey went. So both of us, my co-founder and myself, came from Imperial. We started, after going into industry, we started developing outside of my parents' office, parents' garage, which we converted into an office. And at that time, we had two main data sets. So we're exploring doing bin level analytics. So you walk up to a bin with an item, and then it tells you which bin category to put the item in. And we were also exploring conveyor-based analytics, where it was in a large facility. So you can imagine our folder structure at this time, working locally, was bin dataset and conveyor dataset, which is fine for a team of two hacking in a small office space. So then what we did was we needed some more data. So we took an old 40-pound running machine off eBay. We took away the top bits, and we fitted a load of cameras on top of it. And that was our first kind of conveyor, and that allowed us to get our first proper sets of conveyor and bin data sets. So the labeling then, we thought, well, okay, conveyor is not going to work. We've got time now. So we've got two different conveyor data sets, and we've got two different bin data sets. So we put a date on both of them. And that was it for that time period. So you can see down the bottom left here, there are some images of our data, and we came up with a nice way of automatically labeling data sets. So we could collect large numbers of data sets very quickly, which is great from a machine learning perspective, but not so good from trying to keep everything organized. So then we retrofitted our conveyor and we improved it. We added some lights, which allowed us to get better data. And then we collected more data sets. So we had a think, and we went, what should we label our data sets now? So we came up with a brilliant idea of changing it from conveyor to conveyor old and conveyor new, which gave us 4 data sets, a little bit more messy, but we have some idea still of where the data came from and what it actually is. So then we got accepted onto the Microsoft AI for Good accelerator. We moved offices, and we managed to have enough resources to buy a proper conveyor belt. Not so much to hire a delivery firm though. So we had to rent a van and try and shove it in. So here's our new conveyor. But what happens now to this data set, right? So we've already labeled it old. We can't really do like old, middle, a new conveyor or something like that because it's just not scalable. And also at the time we're deploying things into facilities. So we have live data from facilities, which is taking up a lot of storage on Azure and giving us more data, and also the synthetic route as well, which we're exploring. So we had bins, conveyors, real life conveyors in facilities, and synthetic data. And all of these need managing, which kind of turns into spaghetti. So let's break down the problem. So we have an edge device, which is running in that box that I showed you a couple of slides ago. That runs a bit of code, right? And that's in the production environment. Then we source that code from somewhere, GitHub, and we build an image that deploys onto the edge device. We also want a data science hack space, which I'll call it, which is a non-production zone for anyone that's in data science and just needs to hack around. And also, generally, data scientists don't do production code at the start, right? So you need a bit of time exploring, then you productionize it, and then you go to the end. So in the middle of this, you need something that stops you from sending a model or sending any code into your production environment without it being tested or without you validating the model. So this has been put by Gandalf. Also, the other thing that you need to think of is how are we going to version these data sets? So this was the problem in the last couple of slides of, we've got new, etc., and they're all different data sets. They combine in some way. We may want to combine multiple data sets and train on multiple ones of them. And we also need to keep a versioning here. So if we have a new data set that is slightly improved, or we collect more data from our pipeline at the edge, then that goes into a version data set and creates a new data set. And the important thing here is that whenever you compare models, you need to know, am I comparing this current architecture with the current data set versus an old architecture with the same data set? So you always want to compare the same data set, basically, if you've changed the model. You only want to change one thing at one time. So that deals with the model side. But the other thing is we have a number of modules on the edge, right? So we want to make the amount of it as deployable as possible. So deploy, for example, the camera module differently to the model. And that allows us to really break down the code and make sure that whatever code is running the model is as small as possible, so the other modules can deploy at separate times. And that's what Docker and GitHub Actions allow us to do. So we can build these containers, generally with an API, which contains the model. And then the other containers query that API, and it allows these predictions to happen in real time on an edge device. So here comes along to the answer to the problem on the right-hand side, which is MLflow and Azure Machine Learning. So thinking about how we need to version these data, we need somewhere where we can submit experiments, we can version models, we can version data sets, and also it's easy to pull those new models down onto containers and build them. And that's what Azure Machine Learning allows us to do. So I have, no, not questions. I have a demo, which hopefully you can see the whole screen. Say if not. But what I did was I took YOLO v5, which is in PyTorch, so I will get a bonus point there, and built that into the Azure Machine Learning pipeline. So here is a simple PyTorch model. It runs just object detection, but we want to build this into something that we can use multiple data sets, have a pipeline to deploy, and easily build them into containers. So diving into the code a bit, what Azure Machine Learning allows us to do is we can get runs from, we can use their SDK to allow us to log runs and log the metrics. So in this one, what we do is we initiate the Azure Machine Learning run. We have some config files that allow us to deal with the model and the hyperparameters. So if you were thinking about scaling this, you could change the hyperparameters and deploy them pretty easily. We do some modifications because we want to export it to Onyx format. And then finally, we can deploy the model and read the results. So in these results here, you can see that we're logging the results to Azure Machine Learning. And this basically runs very easily. So you have a configuration file here. We can choose our data set. We have the relative dependencies, which are loaded from a conda file, which is very easy. And then that deploys to the machine learning, to the cluster. So here is an example of one of the data sets that we can version. So instead of just having a simple folder structure where you say, you know, for example, the file name dictates exactly what the model is, we can start moving towards custom tags. So for example, we can say a location where it's collected. So this one should be PyTorch Meetup. The type of images, the type of data it is, so images and segmentation, the width of conveyor that it's taken on, so that we know, for example, if it's coming from a 2-meter conveyor or 1-meter conveyor, etc. And these sort of tags can build up and allow us to version our data in a more sensible and scalable way. The other useful thing with this is obviously anyone can access it in the company. So prior to you know, Microsoft, we were a team that was very small. We were 2 people, and I was the only person coding. Whereas now with scaling, we have more people operating on the data, and having that access and everyone being able to deploy their code is really important. And the other final thing is that you can version your data set. So here, for example, there's version 1, which I just created for this meetup. But if you submit another version and you change your files, you can reversion your data set, and then you keep you keep the accountability of each of the data sets, and you can version them. So here is an example of a couple of experiments that I ran. Didn't really change much, only the number of epochs that there was running for. So this one obviously ran for a lot longer, and this one was much shorter. But you can see, for example, let's go into run 3. The outputs and logs are done here, and that was that logging step that I had earlier. And this allows you to really easily deploy lots of models to the cluster, and you're not constrained by compute power that you have locally. And you can also log those results of each experiment onto the platform. The other thing that's really useful is that you can get output logs. So as part of the script, I used Log Guru to output some logs, and they can output, you know, whether you've passed the file and where the errors are, and some additional metrics, including TensorBoard events and some photos. So for example, you can get lots of photos and information from your TensorBoard logs and download the weights afterwards. Okay, so I think the only thing to go through now is actually how to deploy it. So one nice thing about Visual Studio Code is that there's an extension here, which allows us to then deploy some models to our experiments. So here is an ML Studio running in the cloud, and we can choose the experiment and rerun the experiment from here. So if I go run experiment, then we can choose the compute, which is a cluster, and we can choose the run configuration, and that will automatically submit the run to Azure ML. It's that easy, which is nice. So here is the next run. It's building the container, it will run all the logs, and it will go from there. And just to show you our compute resource, you can set this up here. So this scales as you need it, which is the other nice thing. So at the moment, there is nothing running, but when this run submits, it will initiate a new node, scale the cluster, only run for the amount of time that you need, and then reduce it down, which helps you to save on a significant amount of costs, which, unless you're Google, you kind of want to do. So I think that's it. Any questions from your side?
Okay, and let's start with this. Currently now, what you've implemented is geared towards the industry.
Yes.
Right? So is it possible, without that we've got the machine learning in the cloud, that if the home users have recycling bin, you know, normally they have them colored green, paper, and so on. Is it possible to generate incomes from it by taxing them according to their recyclable items. For example, items like papers that are recyclable, they can get incentive, and things like electronic equipment, metals, and everything, they get charged for it.
Yeah, so we did investigate the bin level side, but there's obviously a privacy problem there. So if you're starting identifying people down to a person level, then that's not so good. But one thing that we did explore was potentially at the corporate level. So you could start looking at things like bin placements, where is the optimal place to put a bin to stop or to increase recycling rates. Also figure out which areas of a particular campus are the most offending, and then you can use that information. The only thing with bins is obviously you need to deploy a large number of devices, and all of those devices need power. And it's quite hard to get power at some bin locations. So I think the battle, if it's to be won anywhere, is either in the facility or actually just increasing the granularity of sorting at home, and not necessarily taxing or policing people from a bin level perspective.
Okay, thank you. So what is the future of your, are you going to expand this beyond what is currently doing?
Yeah, so that was a demo of an off-the-shelf model. We have different models that run. Specifically, waste facilities care about weight data. So from just an object bounding box perspective, it's quite hard to get weight information. So we're going down the segmentation route, which allows you to get more interesting information as to the weight data of a particular item. So yeah, we're expanding both on the machine learning side, but also actually on the deployment side. So things like optimizing the pipeline, building it out so we have better quality control gates for the models we deploy, and bespoke models for each use case.
Okay, cool.
Peter, there is a question in the chat asking about the yellow 5 model. Would you like to address that?
Yeah, sure. So as I said, this is an example one that we ran for PyTorch. As you can see, the Azure ML was pretty sparse. So we have segmentation models that we run. It's the feature of a PyTorch meetup that you need to keep relatively quiet.
So currently now you're using YOLO 5. And you're using Azure as a cloud. Is it possible to make it portable, put it on Raspberry Pi, and when trucks and lorries goes around, then they can collect more samples, and restaurants can start to use these devices, government premises. I'm just looking beyond the factory level, where all this can be, applicable and monetized.
Yeah, so we can run it on very small containers. So the pipeline that I showed you there was the first part of that pipeline, which is the data versioning and the model side. The second part of the pipeline is deploying it to Docker containers and images, and those run on the edge. So we don't compute anything in the cloud, it all runs on an edge device.
Cool stuff. Well done. Good technology.
I had a question. So interesting talk. I liked it. The production, all of the challenges of versioning data and all of these things that you don't get when you just use a existing public data set sounds challenging. I was curious what your thoughts are on, like, is these techniques kind of applicable to things for reproducibility and research? Or how different is that problem compared to the kind of productionalizing data collection stuff?
So I think for research, it just depends on your data problem. So is it an issue where you get lots of different data sets? And if that's part of it, then you need to manage those data sets. And I think this is a really good way specifically of managing data sets and models. When you're simply, one data set and you're going from there, then the only thing that I think this benefits you is if you don't have the local resources to run something and you want the scalability of a cluster to run on, which allows you to scale up and scale down as you go. But you could get that through other means other than this. So you could just spawn a cluster somewhere else.
Cool. Yeah, because I mean, there's lots of different things in the data and data augmentation and all of these kind of hidden behind the scenes stuff that doesn't really get captured in the final paper. Even like different data set splits and data kind of like sampling stuff.
Yeah, I think one really useful thing about this is you can pipeline methods. So say you start with an original data set and you have a method of augmenting that data set, then you can create that in a pipeline so that you have each of these individual modules that run, and then finally train your model on the output. So it's useful for things like complicated pipelines and sort of sorting that out logically.
Interesting. Sounds very cool.
Yeah. And if you look at it currently now, most in recycling industry, sorting is still done by manually. I mean, they still use human being to sort paper, sort plastic, sort this and into different before they actually being recycled. And the same thing in the car industry, when cars are salvaged, pressed, they still have to all these components still have to be sorted manually. Is it possible, your system to be incorporated into automatic sorters, where a robot can use that knowledge you've built and can pick different devices and put them in the different, collections area?
Yes, definitely something. Definitely something that's possible and something that we're working on.
Yes.
So the way that we see it is human pickers in facilities, they have eyes which see the items, right? And then they have the arms that sort them. And so part of the problem is the visual problem, and the second problem is matching that with the arms, the conveyor belt speed, and picking up in an optimal way. So this is half of that, which is the vision side, and then we're working with partners to get the other side.
Cool. Good stuff. Well done. Yeah.
So Peter, I have a very naive question about the data sets. So if you combine all the data sets you have, do you just get better performance, or is that too simplistic?
It depends, because some are for a different use case and some are for different countries, for example. So it depends on what the use case is for. And then so for each different use case, we have different data sets.
So it would be, every model would be fine-tuned for a specific country based on what kind of brands, what kind of waste they have?
Yeah, so you can either do that or you can do a domain adaptation approach. But basically, you don't want significant amounts of data from every country. For example, in France, milk cartons are slightly different to in the UK, and you get a lot of those small kind of round milk bottles, whereas in the UK, you get the very familiar larger ones. And so that's just one example, but there are many examples where, things go slightly differently. Things like the difference between a green bottle. If it's green transparent, there are different shades of that green. So you can have anywhere between it's transparent and it's very green. And depending on even on the facility level or what the person's feeling like in the day, you can sort them in different categories. So some will go in the clear transparent and some will go in green. And it varies very differently depending on use case.
I see.
That's very interesting. Do you see any sort of seasonal or temporal changes in the data as well?
Yes. So lots of PT clear bottles in the summer because everyone drinks loads of water, and those are those sort of things. So you see large variations. You see, for example, at Christmas time, a lot of the plants are on overdrive because everyone produces a lot of waste around Christmas. So not only from the type of products, but also from the load on the plants, it changes quite a lot.
Cool, really awesome stuff. I could imagine you could even get some potential revenue model for your work, as in selling this data to companies who would like to understand what kind of seasonality is there in their product consumption.
Yeah, exactly. Yeah, Recycleye is a company, so we are marketing it. Don't worry.
So Peter, you shared a very nice story about how you guys started and you bought your first conveyor belts. So what's the big vision? Where do you want to be in 10 years?
Yeah, good question. So I think the key is to really democratize or add transparency to the waste industry. So at the moment, you kind of throw that item in the bin, right? And then you have no idea really what happens to it. It goes off, it goes out of your mind, and you kind of blame, or you don't blame, but you pass the buck on to the waste management company, right? And then they have to sort it. And what we're trying to do is add that data and that analytics to the waste so that we can start tracking items, we can start informing brands of how their packaging is performing in these facilities. So some of the sorters that are in facilities aren't, the packaging that goes through, it can't be recognized by the sorting machines. And things like black plastics, things like bioplastics, for example, are also becoming a problem. And having that feedback loop, both from the waste facility to the brands and also to the consumers, I think is really important. So the big vision is to add transparency to the waste industry and just add that data and that knowledge back so that customers and brands can make an informed choice.
Exciting. Well, do we have any more questions for Peter? Please shout out or type them in. And I guess another question would be, what countries are you planning to collaborate with?
So at the moment, we're sticking with Europe. We find obviously travel at the moment is quite tricky as well. So I think we're going to try and stay close to home for a little while and really validate the system here, and then we'll start exploring other countries. But yeah, some countries have better recycling rates than others. The other thing to think about is, which countries are ready for data. So not only, for example, in the manual picking case, a lot of firms, for example, in India or places like that, wages are cheaper than in Europe. So you want to aim for the countries where wages are expensive, and actually, adding this automation adds value. So that's why sticking to Europe for now is a good use case.
I think it all depends on how you weigh it out, because there are quite a few people actually in that pipeline where you are. Some are just also doing recycling by filtering out and then taking things from UK, transporting to other places or bringing back. And some, for example, they are taking materials from here to Germany, where car industry are. And from Germany, they bring other things for building and some other things here. So you probably want to spread out. Of course, you have to look at where monetize, can go. But at the same time, things like NGOs, they'll be much more interested working with you, because now, environmental issues are at large, and all these are caused by waste. People throwing things into the sea, plastic into the sea, and so on. So that could also be something that they can do for dredging and many other things. So it got quite wide areas of applications, not only within the waste or everything, but even the environmental buildings, and all those areas. Council can also, you can work with councils, local boroughs, where they are looking for solutions, at local levels. So there are many other areas that this can be applied to.
Yeah, I think it's really exciting. If you get talent interested in detecting waste and interested in the waste industry and make the waste industry sexy, then we'll start solving some of the big world problems. And I think then it gets really exciting. So.
Look here, I have a mean question potentially. Do you know company Grey Parrot?
Yes, I do.
How are you different?
So we have an aspect of hardware in our system. So Grey Parrot is currently focusing on software only. And the other thing we're concentrating on is very much the deployment side of our system. So it's all well having, small amounts of data that you collect in a sort of old school, like a way at a plant, but it's making it scalable. So having things like Azure allowing us to deploy these models at scale and reduce that cycle of development is really important in your accuracy. So a similar thing has happened in software engineering. So you started with DevOps, and DevOps has really improved the cycles of software engineering. And the same thing is happening with machine learning and data science. And so as you reduce those cycles, you increase your accuracies faster, you develop faster, and you improve your system past what your competition can.
I see. Cool. Well, thank you so much for the talk, Peter. And thank you so much to all of our speakers, to Zach and Artur as well. And in real life, we would have a big round of applause, but here you just have to kind of imagine that. Or we could all unmute ourselves and clap. Thank you so much, guys. And so now is a critical moment because we're going to jump to a different platform. And usually when we do that, we lose at least half of the people, and it always makes me very sad. So let's try this time. We have 30 people on the call. Let's try to keep as many as possible going. So I've just posted a link to networking, which is on Icebreaker.